Okay, so I'm going to talk to you about biased agonism. And really there are two take-home messages which I would like to give you this morning. First one is that 5-HT1A receptor subpopulations have signaling properties that can be pharmacologically distinguished. So I'm going to tell you about drugs, but I'm going to talk pharmacology. And the second take-home message is that novel, selective, biased agonists targeting post-synaptic 5-HT1A receptors show improved therapeutic potential. So those are the two things I'd like to tell you about this morning. Okay, we've seen a kind of schematic of a serotonergic neuron releasing 5-HT. We think that in depression, the serotonergic levels are deficient. So we give often SSRIs to boost the serotonin release, which will activate those post-synaptic receptors, for example, in the cortex, and have an antidepressant activity. This is what we're looking for. We're familiar with this. Of course, we have serotonin being released onto the autoreceptors, which we know is associated with a delay of therapeutic onset. And of course, the other thing is that serotonin hits all of the serotonin receptors, not just those mediating the antidepressant response, but also those that limit the antidepressant response, or those which are responsible for side effects. So just a generalized effect on serotonergic transmission probably is not an ideal strategy to treat depressive states. You would rather target those receptors which are directly involved with antidepressant response, if you can. Okay, so what do people do to try and circumvent this? Well, one of the things, of course, is to give a 5-HT1 antagonist, and this is what we heard about. For example, the DU compound, which blocks the autoreceptors. Okay, so you increase the serotonin levels, you avoid the delay of therapeutic onset, but of course the problem is that it's blocking all of the 5-HT1A receptors, including those which are mediating the antidepressant response. So this is not an ideal solution. Okay, so what else can we do? Well, maybe we should directly target 5-HT1A receptors themselves with a 5-HT1A agonist. And so if you give a 5-HT1 agonist, you will activate those postsynaptic 5-HT1A receptors, which are mediating antidepressant response. But there, it's not ideal either, because you will also hit the presynaptic 5-HT1A receptors. You'll suppress serotonin release, at least until those receptors are desensitized, and we've heard about some of the mechanisms. And of course, you'll activate other subpopulations of serotonin 5-HT1A receptors involved in thermoregulation or neuroendocrine response or memory function. And maybe you'll have some side effects because of that. So if you could choose, really what you would like to have is a biased 5-HT1A agonist. And this would target those postsynaptic 5-HT1A receptors which mediate the antidepressant response, but not activate the other subpopulations of 5-HT1A receptors. This is what we would like to have. Well, the term biased agonism, if you do a literature search in PubMed, you will probably not find very many old references. And the reason is because the concept is quite recent. It only dates from the last 10 or 15 years. And the nomenclature was quite diverse. So, for example, agonist-specific trafficking of intracellular responses, or effector pathway-dependent relative efficacy. So it's quite complicated. And eventually what's happened is that people have converged on this term biased agonism. Okay, so this is what people are talking about now. But the concept is actually quite simple. What we're talking about here is a situation where, unlike classical agonists, which would activate all of the signal transductions pathways available to the receptor, a biased agonist will activate preferentially one or another signal transduction pathways, maybe by preferentially activating different G proteins or effectors. That's the idea of a biased agonist. And of course, if the particular transduction pathway is associated with a therapeutic response or a side effect, then a biased agonist can separate those effects and maybe have a better therapeutic profile. So that's what the concept is about. And if you hunt back in the literature, in this case back to 1994, we can see that there's some signs of that for 5-HT1A receptors. So this was a study where somebody looked at the activation of two different G proteins so G-alpha-I3 and G-alpha-I2, by 4 different agonists. And what they found was that this drug, ipsapiron, showed a nearly 4-fold preference to activate GI3 rather than GI2. And these other drugs didn't. So there was a little indication there that these agonists can preferentially activate one signaling system rather than another. Of course, at that time, we didn't really know what to do with that. But it showed that there was something there. And more recently, there was a study done on RAFA brain tissue showing that 8-hydroxy-D-pad preferentially activated GI3 subunits, whereas buspirone, which is a weak partial agonist, 5-HT1A anxiolytic, activated other G proteins and also inhibited CAMP. So there's indications in the literature that these drugs are not all the same. And in fact, if you hunt through, you can find a whole set of differences between the signaling of RAFA 5-HT1A receptors and hippocampal or cortex post-synaptic 5-HT1A receptors on a number of different parameters. So if you could find a drug which was selective for the receptor and also picked out the signaling pathways which are present post-synaptically in the cortex, perhaps that would be a good antidepressant drug. Now, of course, if I'm standing here talking to you, it's because I think we've got such drugs. And the structures are here. Now, just to say that these compounds, which are recent, these are chemically and pharmacologically different to the old azapirone type, buspirone type drugs, which many of us know about and have a number of limitations because they're not very selective, metabolically they're unstable, and they're only partial agonists. So these drugs have been referred to by some authors as being selective 5-HT1A superagonist. So we were talking about a different category of chemistry and pharmacology here. And in particular, this compound behaves as a post-synaptic preferential biased agonist. And that's the one I'm going to tell you about in the remaining slides. So firstly, to say it's extremely selective. So you see here in the top left, the affinity of 5-HT1A receptors, and basically it's got no affinity at other receptors. Unlike some reference drugs, which you see, for example, buspirone here has some affinity of 5-HT1A, but it also has affinity at D2 and other receptors. So these new drugs are very, very, very selective, and they're also very efficacious in screening tests of agonist efficacy, more efficacious than the reference compounds. But also biased agonists. So this compound, you can see here, has higher potency and efficacy for activation of G-alpha-I than for G-alpha-O. So it's differentiating the signaling, whereas 5-HT here has a similar potency and efficacy for both. So we have a biased agonist at the G protein level, but it goes beyond that, because if you look at a whole series of different intracellular responses, including ERK phosphorylation and as well the G proteins, but also receptor internalization and CAMP inhibition. You can see that each drug that was tested had its own order of potency. So in the case of F15599, it potently stimulated ERK and then the other responses. And you can see it did so at a low concentration. Whereas this drug, for example, elicited all the responses at roughly the same concentration. So each drug had its own signaling fingerprint. Now this is important because we have to be careful now we know these kinds of results, we have to be careful about generalizing about 5-HT1A agonists. We have to say, well, perhaps these agonists are not eliciting the same intracellular responses, and therefore maybe they don't have the same therapeutic potential. And of course, the ERK signaling is interesting because there's a whole series of studies that say that ERK phosphorylation deficits may contribute to depressive disorders. And therefore, if you have a drug which favors activation of ERK, well, that may be a good thing to achieve antidepressant activity. Of course, what I've shown you is just biased agonism in vitro, just on cellular systems. But what happens in vivo? Does this translate to physiological responses in whole animals. And this was data gathered in Francesco Artigas's lab by electrophysiology. And if you give cumulative doses of F15599, you can see that you actually increase the firing of post-synaptic cortical pyramidal neurons, and this is dose dependent. And the lowest active dose is as low as 0.2 micrograms per kilogram. Now, what I'd like you to notice is that if you then test the same drug on the dorsal raphe firing, you have to go to something like 5 or 10 micrograms in order to inhibit the serotonergic firing. So in other words, we have a low dose effect in the cortex, which is increasing the FOHT1 activity in that brain region, but without suppressing serotonergic neuron electrical activity. And we have a similar message from neurochemistry studies. This is microanalysis, and you can see that at very low doses, we stimulate dopamine release in the frontal cortex. So this is a postsynaptic cortical effect, which is blocked by a FOHT1 antagonist. But you have to go to much higher doses to suppress serotonin release. So once again, what we have here is a neurochemical demonstration that we are preferentially activating cortical FOHT1A receptors. And you can see that on the AUC graph at the top right. You can see that the low doses, we only have the cortical effect without any effect on the serotonergic neuron activity. And we can derive ratios of ED50s, and in the case of F15599, we have about an 8-fold ratio of ED50s between the postsynaptic effect and the presynaptic effect in this neurochemical assay. I'd just like to point out that this drug, which is an analog of that one, goes the other way. You notice that F1374 actually has a ratio which is switched. In other words, it's actually preferential presynaptic. So you can have drugs which preferentially activate cortical FOHT1A and other drugs which preferentially activate the presynaptic FOHT1A autoreceptors. And these have different profiles in terms of desensitization. So here the presynaptic FOHT1A agonist rapidly desensitize the serotonin release. So this is the control condition. It suppresses serotonin release. But after just 2 or 3 days of treatment with this drug, well, you lose that response completely because those autoreceptors are desensitized. Whereas with this compound, F15599, we have to give the drug for like 2 weeks at a very high dose in order to attenuate the inhibition of serotonin release. In other words, it's just not hitting the FOHT1A autoreceptors. It's just not going there, except at a very high dose. And we've done other measures just to look at this presynaptic, postsynaptic difference. And this is results on ex vivo measures of immediate early gene expression of CFOS, which is like a marker of neuronal activation. And if you look at F15599, you see that it potently stimulates frontal cortex CFOS expression, but very little in the dorsal raphe. Whereas the presynaptic compound preferentially activates CFOS expression in the dorsal raphe and less so in the frontal cortex. So yet another measure saying that you can target these receptors differentially. So how does this translate to measures of mood control? Well, here are 2 experiments that we did. So this is the top one is the fall swim test. And of course, if you look at some reference compounds such as buspirone, well, there's no activity. Citalopram is not active acutely. If you give an SSRI, you have to administer it repeatedly in order to see an inhibition of the mobility in this test. Now, flizinoxane, which is a partial agonist of FOHT1A, has a little bit of activity. But look at these drugs. These completely suppress the mobility. Now, that's pretty impressive because these are acute treatments. So a 5-HT1A agonist, if it's efficacious and potent, will completely normalize the behavior of the animals acutely. And this is maintained over chronic treatment. But what's surprising here is the fact that these curves are superimposable. Now, the reason that's surprising is because there's a difference in affinity between those 2 compounds. F15599 is markedly less the affinity is markedly less than that of F13714, you would expect the curve to be here, according to the binding affinity. But in reality, what you observe is that the curve is shifted to the left. It's actually more potent. And we think the reason is because it has a preferential post-synaptic activity. I should just mention that the differences are not due to differences in bioavailability, but they're both very highly bioavailable. They both penetrate the brain very effectively. So we're not looking at a PK difference. Okay, this is a pharmacological activity difference. And so the post-synaptic 5-HT1A agonist is very potent for eliciting the antidepressant-like response. And we see the similar kind of pattern on another test, which is the stress-induced USV, where the reference compounds are active. And once again, we see almost a superimposed effect of the two biased agonists. So post-synaptic targeting of 5-HT1A improves the therapeutic profile. Now, of course, if we're talking about 5-HT1A in the cortex, and we're talking about increased dopamine release in the cortex, then, well, immediately you think of cognition. You think, well, if you're increasing cognitive function, that would be a good thing if you could achieve that. And so we did some memory tests using a whole board paradigm. And a whole board is very simple. It's just a box with a series of holes, and some of the holes have a food reward. And the rats learn very quickly to find the food reward. And we can score that, and therefore attribute a working memory measure. And the control animals are here at the top. They have high scores. But if you give them a compound such as phencyclidine or PCP, you'll disrupt cortical function, and therefore you will reduce those scores. And here the PCP-treated animals are at the bottom. So what I'd like you to notice is that if you give this biased agonist, you attenuate that cognitive deficit. You attenuate the working memory deficit which has been elicited by PCP. That's because you're favoring cortical activity. The drug by itself doesn't have any effect. But if you look at the presynaptic 5-HT1A agonist, which suppresses serotonin release, well, by itself, it impairs working memory performance after a few days. So in other words, you want to go for the postsynaptic 5-HT1A in the cortex and not for the presynaptic 5-HT1A activation. And we looked at another test, which is a reversal learning task. It's quite complicated, but it's a measure of cognitive flexibility. And PCP induces a deficit in this test. And once again, here are the control animals. They perform well. The PCP-treated animals show a sizable deficit. But the animals receiving the postsynaptic agonists have an alleviated performance. So you partially rescue the deficit. Whereas the the presynaptic biased agonist, well, it certainly doesn't attenuate, and it may actually make things worse. Okay, just in the last minute, last data slide here, just to say we've actually labeled this drug with F18. This was work done in Luke Zimmer's laboratory, who's present here today. And these results were really interesting because he generated some micro-PET data in rat and in cat. And what I'd like to point out is that the labeling was higher in the cingulate cortex than in the hippocampus. Here, this is the background cerebellum. So we have a higher labeling in the cortex than in the hippocampus, which is remarkable, because the density of 5-HT1A is higher in the hippocampus. And yet the drug doesn't label the hippocampus as much as the cortex, because it wants to go to the cortex. It's a cortex-directed or a biased agonist with a cortical preference. I'll skip this slide. This is just to summarize to say that we've got a lot of data now on this compound, so I think it's a pretty interesting story. And just to conclude, we're looking at innovative biological mechanisms. I think we have now moved to biased agonists targeting subpopulations of 5-HT1A receptors, for example, in the cortex. We've got a promising chemical lead, which is acting as a kind of prototype. We can also associate this with a patient stratification strategy. Wouldn't it be nice to associate a biased agonist, cortical agonist, with a strategy whereby you go to those patients, which Paul told us about earlier on, which have a deficit of cortical function because they're expressing this, this polymorphism, the RS6295 polymorphism, which impairs cortical function. So then you would match the right drug with the patients that could most benefit from it. And there's a number of companies that are looking at biased agonists in their research programs. And Neuropsychopharmacology, which is where I'm working, we're actually synthesizing and moving towards developing these novel 5-HT1A biased agonists. I'd just like to acknowledge all the people that contributed to this work, which took place over a number of time, a large amount of time in different laboratories, and I'd like to thank you for your attention.
Thank you very much to Dr. Newman-Tancredi. We do have time for a few questions.
I think you were referring to results from Francesco Ortega's. Actually, you were showing in the frontal cortex that you had an increased activity. Well, generally, the 5-HT1A receptor is associated with an inhibitory transduction system. So how do you explain these increase in firing activity? And I guess this is how also you explain the procognitive effects.
Right. Okay, so the question is, if you've got an inhibitory receptor, how come we've got an increase in electrical activity in those neurons? Which is a good question. And it actually raises, I think, another level of complexity, because 5-HT1A receptors in the frontal cortex are located on different kinds of neurons. They're located in pyramidal neurons, which are glutamatergic, but also in GABAergic neurons, and which are interneurons. So there's two populations of 5-HT1A receptors in that structure, and Francesco Ortega has looked at this in considerable detail. And basically, it's the balance of activity of those two populations which determines the final outcome. So I think maybe a future story, maybe we need to look out for this in the next few years, is that not only will we be targeting 5-HT1A in the cortex, but 5-HT1A in the cortex on pyramidal neurons or on GABAergic interneurons. So I think we can go even further in the specificity of the targeting.
If I may add something, actually we showed that 8-hydroxydipide, we still do not know about this compound F15599, but 8-hydroxydipide activates preferentially at low doses 5-HT1A receptors in GABAergic neurons, and we published that recently in cerebral cortex. So clearly, I think that so far we had 2 bags of 5-HT1A receptors, the pre and the post-synaptic bag. So I think that we have to open the post-synaptic bag in different bags, which may have also differences in terms of agonist activity, coupling mechanism, so on and so forth. Okay, but I wanted to ask you a question, Adrian. According to everything in the field, one would suggest that the best antidepressant compound would be along these lines would be a presynaptic 5-HT1A antagonist and a full agonist at postsynaptic 5-HT1A receptors. I remember some old compounds from Servier, I don't remember what number, they were exactly the reverse. They were full agonists at presynaptic 1A receptor and antagonist at the postsynaptic receptor. Do you think, from your knowledge of medicinal chemistry, do you think that such a compound, I mean, an antagonist, the presynaptic and agonist at postsynaptic receptors could be feasible?
Well, why not? I mean, 5 years ago, we would have said that this kind of drug, was not possible. And here we are presenting, and we're looking at targeting subpopulations of 5-HT1A receptors. So why not? Actually, just to add another comment, F15599 at low doses does not activate the autoreceptors. This is what I've shown you today. Now, I don't know what it does in the presence of a presynaptic agonist. I mean, the drug's there. I mean, it's in the brain. So what's it doing on those autoreceptors? Is it just not binding at all? Or if it's actually binding to the receptors, is it just not activating? Or is it actually blocking something? Now, that's a question we haven't looked at, but would it be really interesting to give a presynaptic agonist and to give 15599 at a very low dose and see if you actually block that? So maybe we already have a drug which combines postsynaptic activation with presynaptic antagonism. But why not? Yeah, sure. I think it's opening up a whole set of questions here, because we're kind of subdividing